Hello, everyone, and thank you for joining us today. My name is Rafal Jingels, and I'm going to be your moderator for today's webinar. I am the Field Application Sciences Director of Analytics at Chain Labs. And so today we'll have Keith, our amazing FAS based in Boston, kind of walk us through a very interesting topic on the Uncle called the amazing blue shift, right? And so what he'll be doing today is essentially tell us about the molecular and biophysical basis of blue shifts, give us examples, but also demonstrate how the Uncle tools are uniquely suited to find and characterize blue shifts, okay? And so we'll have a Q&A session at the end. So if you have any questions, I'm going to recommend that you look at the bottom. There's a Q&A icon. Just simply click on it and type in your questions, and we'll answer them at the end of the presentation. And so now I'll hand it over to Keith, and here we go.
Well, thank you, Rafad, for those kind words. As Rafad indicated, I am the Boston area, New England, and upper Midwest Unchained Labs field application scientist. And today, as Rafad said, we're going to be talking about the amazing blue shift, or what full spectrum fluorescence data can tell you about your protein. And one of the main reasons to be discussing this topic is that it really highlights the utility of our Uncle instrument at doing really full spectral molecular analysis, and it gives you amazing data that I think you'll find highlighted by this talk. In this particular representation of a blue shift, we're representing it as a superhero, and blue shift sounds like a superhero, but of course it isn't. What superheroes and blue shifts have in common is that they're remarkable. And today we're going to spend about a half hour remarking on blue shifts and how interesting they are from a molecular and biophysical basis. So before we get into this, let me just introduce the Uncle instrument. It is an amazing instrument for biophysical characterization. It really is a Swiss Army knife for biophysical characterization. It has a 266 nanometer laser, a 473 nanometer laser, a 660 nanometer laser, an avalanche photodiode, a CCD, and a Peltier element. And when you put all of that together in a single instrument, you have an instrument that can really give you a full suite of biophysical tools to allow you to analyze the thermodynamics as well as the colloidal stability of your protein analytes. Uncle allows you to do full spectrum fluorescence, so that's what is used to uncover the melting points and the onset of melting. Uncle also gives you dynamic light scattering, which will provide you with the hydrodynamic diameter as well as the polydispersity of your molecules. Simultaneously with intrinsic fluorescence, Uncle also does static light scatter, so you can understand your colloidal stability as well as your thermodynamic stability as a function of temperature and time at the same time, contemporaneously. So it's a very powerful instrument. And of course, Uncle, in order to do this, has a Peltier element where you can have a temperature ramp from 15 to 95 degrees and change that ramp rate. You can go for a very slow ramp rate, as 0.1 degrees per minute, or you can do a much faster ramp rate, 1 degree per minute. So by having all of these tools available to you, you can really decipher much about your protein, its thermal stability, its colloidal stability. You can do formulary screenings as well as sequence optimization using the Uncle instrument very easily. It's a really wonderful tool. And the data that we're presenting today all comes from the Uncle instrument. So to begin our conversation about blue shifts, we first have to understand what is fluorescence. And I think this is a really elegant way of saying it. It's emission of light by a substance that absorbs light or other electromagnetic radiation. So it absorbs this radiation and then it emits a photon, and that's what we refer to as fluorescence. Specifically, what we're talking about is a change in orbital. So an electron is moved from a lower energy to a higher energy orbital by the electromagnetic radiation. And when that electron goes back to its natural orbital, it loses energy and it gives that energy off as a photon. When specifically we're talking about protein and UV light, and that's the focus of today's conversation, we're talking about pi to pi star transitions. So the orbital goes from a pi orbital to an electron goes from a pi orbital to a pi star orbital. And when it relaxes back to its normal state, it gives off a photon. And much about the characteristics of that light giving off really informs about intrinsic fluorescence. And that's what we're going to talk about today. So what fluoresces? Well, things with, at least on this level, the things that fluoresce have conjugated double bonds. So proteins, and we're going to be talking about them, have aromatic residues which provide it with fluorescence. But many other molecules which we're familiar with, the fluorochromes fluorescein and rhodamine certainly have conjugated double bonds, and they're useful for fluorescence analysis. And the polyene macrolides, and in the name polyene, you get the idea that there are multiple double bonds here. Amphotericin, which is a potent antifungal, is fluoresces very nicely. Nystatin is also used as an antifungal and it also fluoresces. And the cholesterol binding dye, philippin, which is used to map cholesterol in the membranes, or ergosterol when you're talking about fungus, in the membranes of cells, also fluoresces, and they fluoresce because they have conjugated double bonds. Well, when we're talking about proteins, we're talking about the fluorescence of aromatic amino acids. Specifically, we're talking about tryptophan, which has the highest quantum yield, but actually the lower energy emissions profile of the aromatics. Tyrosine, which has a lower quantum yield, perhaps a half order, an order of magnitude lower than tyrosine, tryptophan, and it has slightly higher energy emissions. And then we're also talking about phenylalanine, which has the lowest quantum yield, but has pretty high energy emissions. And the contribution of phenylalanine typically to an overall protein fluorescence might be minimal unless there are no tryptophans or tyrosines. And this is important. Before we talk about how UNCLE resolves fluorescent signatures, let's talk about the structure of the aromatic amino acids. So tryptophan has an indole, so it has not only the benzene ring, but another double bond, and it has strong fluorescence, a very large quantum yield. Tyrosine, which is pictured on the right of your screen, has an hydroxyl group, and it has moderate quantum yield from its fluorescence. And then phenylalanine, which has an undecorated benzene ring, really has very weak fluorescence with regards to the level of quantum yield from tryptophans and tyrosines. So the actual structure of the amino acid itself plays a huge role in the characteristics of the fluorescence that you receive from those amino acids. With regards to the fluorescence emissions profile, these are all normalized so that the quantum yields appear very much the same. But you can see that tryptophan residues, the emissions profile, the fluorescence emission profile, is centered a little bit to the right of 350. So that's a little less energy than these other emission profiles from the other aromatic amino acids. On the other hand, tyrosine is centered closer to 300. So it has a lower quantum yield, but higher energy, lower wavelengths emissions. And then lastly, phenylalanine has the lowest quantum yield, but again, it's slightly to the left of tyrosine. And again, that represents higher energy emissions from phenylalanine. So each of these fluorescent amino acids contributes something different to the fluorescent profile of your protein. UNCLE does intrinsic fluorescence better. And why does it do it better? Well, typically, if you do an excitation scan of your aromatic amino acids, you'll find that you get the most excitation at 280 nanometers. And many instruments are geared to excite at 280 nanometers. But if you notice, the phenylalanine, which is at the very bottom of the left-hand panel, will not be touched by a 280 nanometer laser. So if your protein doesn't have tryptophans or tyrosines, and it has abundant phenylalanines, a 280 nanometer laser is not going to excite that amino acid, and you're not going to get any emission spectrum. The second thing is, if you notice on the right-hand panel, it's very easy to see that the emissions profiles are cover 280 nanometers. So if you're exciting with a 280 nanometer laser, all the emissions obviously are going to be of a lower energy and are going to be to the right of that higher wavelength. So you're going to miss some of the spectrum. All right, so that's a little unfortunate. Well, if you use a 266 nanometer laser, you can capture that whole spectrum. So it'll excite phenylalanines and allow you to capture the whole spectrum. In addition to that, we use the 266 nanometer laser to also do small particle static light scatter. So in addition to getting intrinsic fluorescence, you get contemporaneous light scatter, and you can really understand the relationship between thermodynamic stability and colloidal stability all in one fell swoop using the Uncle instrument. So it's pretty ideal for doing what we need to do. It's not moving. Oh. There are three things that affect the fluorescence emission of a protein. It's the aromatic residues itself, but it's the emissions profile of those aromatics, and we just discussed that they are different. Not all the aromatics are the same. But also that affects the emissions profile of an aromatic is the degree of solvent exposure. So solvent exposure has a stabilizing effect on that pi star electron, and consequently, when it falls back to its normal state, it gives off lower energy or higher wavelength emissions than it would if it was buried in the hydrophobic core of the protein, where most of the time these aromatic residues live. So there's occasional solvent exposure, but most of the time the aromatics live in a hydrophobic space. But when they are exposed to solvent, you get a change in emission profile, and that change is usually to lower energy, higher wavelength emissions. And we refer to that typically as a red-shifted emissions. There's also quenching caused by nearby amino acids. So for instance, tryptophanes can often quench tyrosine residues when they're in close proximity to one another, and the carbonyl portion of the amide section of the protein backbone itself can cause quenching of aromatics. So in a folded protein, these amino acids are in close proximity to one another, and quenching is definitely a phenomenon that occurs. Before we get into what occurs during denaturation, I do want to point out that I recently looked at a protein paper where they looked at about a dozen proteins that have only one tryptophan residue, and the emission spectrum released by that one tryptophan is vastly different from protein to protein, because each of these tryptophans has different exposure to solvent as well as different quenching effects that may be occurring due to the protein backbone, and consequently the emissions profile, the quantum yield, and the actual spectrum released by each of these tryptophans is different. So the environment of these tryptophan residues of the aromatics themselves really affects the emissions profile that you're going to get from each of the individual amino acids and the meta effect of the emissions profile of the protein. So what happens during a thermal denaturation? So during a thermal denaturation, let's say you're heating a protein from 15 to 95 degrees using a 1 degree minute ramp rate, what you're going to see is the protein starts unfolding. And typically what that means is that those aromatics that are buried in the hydrophobic core of the protein become more solvent exposed. Consequently, as they become more solvent exposed, you have a change in the emissions profile. And typically what happens is you have a diminution of signal, the quantum yield goes down, as well as you have lower energy emissions, higher wavelength emissions, and we refer to that as a redshift. So as the protein unfolds, the aromatics become more solvent exposed, and the result is a diminution of fluorescence signal as well as a redshift in fluorescence signal. And this cartoon clearly illustrates that result where you have a change to the right in the center of the peak. So it moves to the right as it red shifts, as that emission profile changes because the aromatic residues are becoming more solvent exposed. That's very typical. Here's another nice example of a redshift. This is a monoclonal antibody, and many of us work with monoclonal antibodies, so this is a particularly interesting example. You can see as the protein is subjected to a thermal ramp, the fluorescence signature changes, it moves in the right direction, red shifts, and there's a diminution of the quantum U. And this is very typical of what you see in probably about 90% of the proteins that you'll look at. You're going to see a diminution of signal and a red shift in the fluorescence spectrum. Here is an example of using bovine serum, bovine IgG that was heated from 15 to 95 degrees at a 1 degree per minute ramp rate. And the thermogram is displayed as temperature versus barycentric mean. When you plot this out, you can easily see a very apparent red shift. The protein, the barycentric mean goes from around 141 nanometers of light to about 153 nanometers of light. And consequently, when you plot this out, you can see a transition. You can point to the midpoint of that transition, and that's the point where half the molecules are denatured and half the molecules are still native. And we refer to that as the melting point of this protein. And this data generated on the Uncle, very easy to demonstrate the melting point of bovine IgG using the Uncle instrument and many other proteins as well. And this pattern is not only for polyclonal antibody use. In this case, you'll find this pattern is typical not of only of polyclonals, but of monoclonal antibodies as well. The environment of that aromatic residue matters a great deal with regards to the emissions profile. And here's a pretty clear example of changing the environment around an aromatic can alter the emissions profile. So in this particular case, we have pure tryptophan in solution, either in water or in 90% methanol. As you can see, when the tryptophan is in the more hydrophilic environment, it has a profile that is to the red side of the spectrum versus when that amino acid is in 90% methanol, which creates a more hydrophobic environment and causes that tryptophan emissions to shift to the left to more of a blue shift. So if the aromatic goes from a more solvent exposed or more hydrophilic environment to a more hydrophobic environment instead of the other way around, you'll see a resulting blue shift, higher energy emissions, and it will move the curve to the left of the spectrum. And that's what we call a blue shift, and it is pretty phenomenal to see this spectrally. Blue shifts are not common, but there are some proteins that demonstrate very large blue shifts that we're all pretty familiar with. So in this particular case, we're looking at bovine serum albumin. Albumins in general are very common proteins. They're very sticky proteins. They're basically molecular mops, so to speak. And if you're purifying proteins from blood or plasma or platelets, you have to spend an awful long time making sure you don't get cross-contamination with albumin. So bovine serum albumin, when you subject this on the Uncle instrument from to a 15 to 95 degree temperature ramp using a 0.5 degree per minute ramp rate, and you plot these data as BCM, barycentric mean, you can see on the thermogram very easily demonstrated a blue shift. So it's going from 347 nanometers to around 342, 41 nanometers, somewhere between that. That is a a real change in the blue shift direction of the spectrum. And it's very easy to see that there is a left movement of the spectrum to the more blue side of the spectrum, higher energy emissions. Now, you can also see this very easily spectrally using Uncle tools, and you can see that the midpoint at the start of the thermal ramp is to the right of where it ends up. So this is a very clear demonstration of a prominent blue shift in a very common protein. Another way to look at this is that we know that the blue shift is occurring maximally between 15 and 70 degrees. That's where the blue shift is maximal in BSA. And we can plot on the Uncle using Uncle software very easily the starting spectra and the ending spectra. And we can plot a straight line down the middle, and you can see that the ending spectra at 70 degrees is much to the left of where it begins. Again, this is what we refer to as a blue shift. Now, what causes this blue shift in bovine serum albumin? Well, let's look at its molecular structure and try to understand this not from a biophysical, but from a biochemical point of view. What's going on here? So BSA only has two tryptophanes. It has a tryptophan 212, which is in the hydrophobic core of the protein, and it has a tryptophan at 134, and that is much more solvent exposed, as you can see from this diagram of the three-dimensional structure of BSA. And nature has provided us with a really good natural experiment by which we can test what the roles of these two tryptophans are in the fluorescent emission profile of BSA. Unlike BSA, human serum albumin only has one tryptophan. Its tryptophan is only in the hydrophobic core of this protein. So it lacks that more solvent exposed tryptophan that BSA has. So how does HSA differ from BSA with regard to its melting profile? So here we're looking at BSA on the left and human serum albumin on the right. They were both exposed at the same time to the same temperature ramp from 15 to 95 degrees at a 0.5 degree ramp rate. And as you can see here, it's very clear that BSA demonstrates a beautiful blue shift, while HSA does not. HSA has a really prominent red shift without the blue shift that BSA possesses. That suggests that that surface exposed, that solvent exposed tryptophan is playing a huge role in the blue shift emissions profile of bovine serum albumin. But human serum albumin has also been demonstrated to have a small shift, and this was demonstrated by using a chaotropic salt, in this case guanidine. And you can do these sorts of experiments using the Uncle instrument. You can certainly use chaotropic salts and look at transitions using these salts. So HSA has a small blue shift, not quite as prominent as BSA, but certainly has been reported in the literature. And how is this explained? Well, in this case, the tryptophan, the buried hydrophobic tryptophan, which is in domain 2, during denaturation in the chaotropic salt, actually becomes closer to domain 2A, which is even more hydrophobic space than domain 2. So consequently, the internal tryptophan moves from a hydrophobic pocket to an even more hydrophobic pocket, producing a blue shift in a chemotropic salt, chaotropic salt. So what have we learned about blue shifts? Blue shifts can occur from an aromatic residue moving from a more hydrophobic to a more hydrophobic space, whether that is a solvent exposed tryptophan moving to an internal space, or even a buried tryptophan moving to a more hydrophobic space. Both of those can result in blue shifts. Moreover, we learned that blue shifts can occur by thermal or chemical denaturation, and either of these techniques is appropriate, but one may bring out certain types of blue shifts better than others. So you may want to use both if you really want to dig down deeply into the transitions that are occurring in your protein and whether or not there are blue shifts. And again, both of these techniques can easily be done on the Uncle instrument. Let's talk a minute about antibodies, because most of us work, many of us work on antibodies, very common for drug development, for the potential for vaccines. So antibodies are very important molecules, and we want to talk a little bit about the potential of antibody blue shifts. So antibodies at the end of the Y shape have variable domains, and at the very end of those variable domains are complementarity determining regions, or CDRs. And it is those amino acids in the CDRs of antibodies that interact with antigen. So consequently, these amino acids are always, to some degree, solvent exposed, because otherwise they could not interact with antigen. These are very key amino acids in the structure of the antibody. And this whole top region of the antibody can be cleaved, and we refer to that, the whole top region, as the FAB, and then there's the constant region, or the FC of the antibody. So these terms are important for talking about what we're going to talk about with regards to antibodies. So if you look at the CDRs of antibodies and look at the amino acids that are represented there, we notice that there's an overrepresentation of both tryptophanes and tyrosines. So we know that the frequency that amino acids occur in proteins in general, we know the frequency that specifically of tryptophanes and tyrosines, and we know that in the CDRs, these are overrepresented versus their how much representation they have in general protein structure. Now, this is an image on the right that from a paper by Collis et al. That looked at an antibody that recognized virus, and they found that it had an overrepresentation of tryptophanes and tyrosines. But they also looked at antibodies that recognize carbohydrate, lipid, hapten, and protein, and all of them were the same. There's an overrepresentation of tryptophanes and tyrosines in the CDR of antibodies. Now, this cartoon illustrates in composite some of the observations I've made looking on the Uncle at various antibodies that demonstrate typical 1 nanometer or less blue shifts. So blue shifts do occur in antibodies. And what I'd like to propose to you today is that because antibodies go through multiple transitions as they denature, typically 2 or 3 transitions, sometimes the FAB fragment melts cooperatively with the CH2. But sometimes it does not, and the FAB melts all by itself. And I'm going to suggest that sometimes all of those solvent-exposed aromatics, if the FAB is melting by itself, may contribute a great deal to the fluorescence emissions, and those solvent-exposed aromatics falling into a more hydrophobic space may result in a blue shift of antibodies if there's an early transition FAB alone melting. So you might find that in some of your antibodies on occasion. So blue shifts may occur due to solvent-exposed aromatics moving to a hydrophobic space during domain melting. Now, I'm also going to suggest that if the FAB is melting with this CH2 domain, that you may not see this phenomena because all of those internal aromatics are going to weigh heavily on the overall fluorescence emissions profile. But if the FAB is melting alone, you might see a change to a blue shift because of all those solvent-exposed aromatics. There are yet another reason for blue shifts, and that may be unclenching. So as I mentioned earlier, tryptophanes quench tyrosines all the time when they're in close proximity. It's a phenomenon that has been observed many times, but also it's been reported that the carbonyl part of the amide group of the peptide backbone may also be playing a strong role in fluorescence quenching. And again, when the molecule is folded in its native conformation, these amino acids are fairly close together, and quenching is a real phenomenon. But as they unwind, these amino acids move farther apart, and quenching relaxes, and consequently, you can get a large increase in the quantum yield of aromatic residues when they are unquenched. Here's an example of that produced on the Uncle instrument. So here is a protein that was heated from 15 to 95 degrees using a 0.5 degree ramp rate. We're only showing the part of the thermogram from 40 to 95 degrees because that uninformative part of the spectrum was not necessary to show this point. And in this case, as opposed to using VCM, we're actually plotting the change in fluorescence using area under the curve, essentially emission spectrum area analysis. And that's a very sensitive technique for looking at quantum yield. And because Uncle allows you to analyze fluorescence using a number of different tools, we can really capture this increase in quantum yield. So at around 60 degrees, you can see here that there's a huge change, and that's the onset of melting, a huge change in the area, suggesting a dramatic increase in quantum yield as this protein is melting. And that suggests a real unquenching phenomenon. And you can look at that by looking at two spectra. The phenomena really occurs between 60 and 70 degrees. You can see that both on the image pictured on the left as well as the one on the right. And you can see here very clearly that there's a huge increase in quantum yield from 60 to 70 degrees. But you can also notice that this unquenching changes the profile slightly rightward. So this would suggest that the unquenching in this case is due to tryptophanes being unquenched because the spectrum that's resolved looks more rightward, more tryptophan, more centered around 350 nanometers, as opposed to tyrosine, which would be to the left of that. But in this particular movie, we can see a wonderful example of unquenching of tyrosine residues. So in this particular case, the signal from tryptophanes is going down during a thermal melt from 15 to 90 degrees, while the signal from tyrosines is increasing dramatically. And this would also result in a prominent blue shift. So you can have unquenching of tyrosines leading to a blue shift. So there are several phenomena which can lead to blue shifts. So blue shifts may be caused by the unquenching of aromatics during melting. So the change in the position of the aromatics from a more solvent exposed to a more hydrophobic space can result in a blue shift. But also unquenching of tyrosines during melting may also result in a blue shift. Okay, so now that we've looked at blue shifts and we understand what blue shifts are from a biophysical and a biochemical basis, let's talk a little bit about the Uncle and how Uncle allows you to analyze blue shifts. Some of you may have an Uncle and you can go right back to your Uncle instrument and start analyzing blue shifts. Some of you don't have an Uncle, and what this will be is a small primer in how user-friendly Uncle software is with regards to looking at blue shifts. So here we've highlighted BSA, a protein that undergoes a prominent blue shift, and you highlight that protein using the Uncle software. And if you right-click on your mouse and ask it to display the differential equation, it will. And as you can see in this differential equation, there's a very prominent inflection point. And it's very easy to see that that would correspond to the melting point of BSA with regards to its blue shift. You can also use our advanced analysis menu, and you can push on the button to the right of the TM after the protein's highlighted, and you'll see the option to move from a peak analysis mode to a trough analysis mode. So now the software is automatically not looking for red shifts, but is looking exclusively for blue shifts. If you hit the recalculate button, now the software will report on blue shifts. And some of those blue shifts you'll be able to see very easily by eye, but some of those blue shifts are much more subtle, and using the analysis tool will bring out blue shifts that you might have missed. Again, if you can ask the software to put a TM line. You can do this whether you put the differential equation yourself and put a TM line there, or the software will automatically drop a TM line at the inflection point of the differential equation, and that will report on your TM of your blue shifted proteins. So what have we learned? Well, we've learned quite a few things today, I think. I hope so. We've learned that intrinsic fluorescence is a product of aromatic residues, their exposure to the solvent, and their degree of quenching. All of that together gives the fluorescent profile of a protein. As a protein melts, aromatics typically become more solvent exposed, but occasionally less so, and this will result in a blue shift. Blue shifts can also occur if unquenching of tyrosine residues is predominant. And probably most importantly for today's webinar is that Uncle's full spectrum analysis helps detect and unravel the molecular and biophysical basis of blue shifts. Well, I want to thank you for your time. It's greatly appreciated. If you want to learn more, you can email info@unchainedlabs.com and your questions will be answered. But you can also email me directly, keith.solomon@unchainedlabs.com, and I'm happy to answer emails regarding anything with regards to the Uncle instrument, fluorescence, or blue shifts. I'm happy to participate in a conversation with you. So once again, thank you for your time, and I believe we have time now for some Q&A.
Excellent. Thank you, Keith. So let's see. So we have a few questions, but thank you for a wonderful presentation. I really enjoyed this. I think it's a topic that we don't cover as much, but it offers really interesting insight into the molecular basis for some of the things that we see, but also just kind of shows you how flexible the Uncle is. So I thought it was quite relevant and quite good. So we have some questions. I'm going to start with the first one, which is, do antibody blue shifts suggest anything about antibody stability or affinity?
Not that I'm aware of. Unfortunately, not many antibodies demonstrate blue shifts, but it may be that there is something about antibodies that do that might affect developmentability, formulation. It's an interesting question that requires further research, and hopefully we can really start teasing this apart a little bit more, because there may be something unique about antibodies that demonstrate blue shifts that may affect how they're produced, their thermal stability, their chemical stability, and their long-term shelf life that we really have to get a handle on.
Sounds good. Thank you. So there's a question that came via the chat and also one that was submitted before, but they seem to be related. So Camille Dagaye asked a question, which was essentially, does the presence of a blue shift inform you about either the thermodynamic or colloidal stability of your protein, right? In other words, are we looking at aggregation, or is it something else that's happening that's causing that?
Well, again, this is related to the first question that you answered. I don't think we really know the answer to that, certainly not completely. Obviously, one thing that you can glean from blue shifts is that there may be solvent-exposed aromatics. Mm-hmm. And solvent-exposed aromatics tend to be very sticky. So it may be hard to develop a protein that's particularly sticky, because it may bind to other proteins very easily. It may be hard to purify. It may affect its formulation as well. So there are some clues about molecular structure that comes from understanding blue shifts, but it's not quite clear enough to me whether you can really say, well, this has a blue shift, so consequently, we must use a histidine buffer to formulate. I don't think we're at that level in any way, shape, or form.
Okay, that's fair. Let's see. So does formulation impact blue shifts?
Yes, so formulation has a strong effect on the thermal profile of a protein. Its melting behavior is greatly influenced by formulation. the presence of salts, the presence of amino acids. You can imagine instances where addition of amino acids in a formulation may have a quenching effect on the amino acids that are in the structure of the protein, the aromatics. You may also, and of course, the melting behavior is strongly impacted by the formulation. So I'm going to say that yes, blue shifts will be affected by formulation, though whether or not they completely disappear in some formulations or are more prominent in others, I can't really say for certain, but I would suspect that that would be the case.
That's fair. And I think, and as we'll see in part 3, right, we have the SLS detector. And so certainly an Uncle user could use SLS to assess aggregation if they were concerned about that. So I think that's an additional benefit of the platform.
The contemporaneous gathering of both thermal denaturation information and colloidal stability is a key feature of the Uncle, which allows it to be a very powerful tool, because if you don't understand aggregate behavior, at the same time you're understanding thermal stability, you really haven't captured the full picture.
Yes, yeah, absolutely. So we have time for one more question. Let's see, what else do we have going on? Okay, so one is, so it looks like it's a two-part question, but are there any proteins that can essentially be used to demonstrate blue shifts on the Uncle? And can you tell us exactly how the Uncle is better suited to do that?
All right, so one very common protein, easy to buy, you can open up your Sigma catalog and get it tomorrow, is bovine serum albumin. And that's probably for teaching purposes, a great protein to demonstrate a blue shift. And the reason that Uncle allows you to understand blue shifts very well is that a number of features of the hardware and the software. First of all, it gives you the full spectrum analysis. So you have a 266 laser, and consequently, you're not truncating the emissions that you're seeing from your aromatic residues, which is going to allow you to get an overall better handle on any changes in fluorescence that may occur as a product of temperature. In addition to that, Uncle software tools allow you to look for both red shifts and blue shifts very effectively. You can look at them by eye, by looking at differential equations, which the Uncle software will put right on top of your thermogram, as well as using the automatic tools, trough selection or peak selection, which will allow you to differentiate red shifts and blue shifts very easily. Plus, you can look at the spectrum of the protein. You just don't get a thermogram. You can really dig down in that spectral analysis, and you can create the kind of movie where we just looked at that the tryptophan signal goes down while the tyrosine signal goes up. Really a beautiful example of a blue shift, and makes for an exciting slide in a presentation. So all of those tools are available on the Uncle instrument, and I'm very glad to be able to use that Uncle instrument to create the slides for today's talk.
Excellent. Thank you so much, Keith. We appreciate it. And thanks for answering the questions, but also thanks for a really great and insightful presentation. I think, again, it's a really interesting topic, and you know, we hope to hear more about it. I also want to thank everyone for attending today. You know, we really appreciate your audience. And so if you have any questions, if you'd like to further explore any of the topics covered today, we encourage you to follow up with questions. I think Keith submitted his email and his contact information, but you can also reach us at info@unchainedlabs.com, and that will make its way to Keith or to any of us, and we'll happily answer your questions. I also want to take the time to encourage you to register if you have not done so for part 3. As a reminder, this is a 3-part series. And so in the next session, Kevin, our director of marketing, will essentially go over the other aspects of the Uncle, which offer you a complete picture, if you want, when it comes to characterizing proteins, developing formulations, and matters of that sort. Okay. So thanks again to everyone, and have a good one.
Thank you.
Cheers.